So I think it's not going to be that you could just throw in this one enzyme and it's going to fix everything. However, there are compounds that are stimulatory for repair processes, and maybe these might actually work.
So hello and welcome to the Sheekey Science Show, where in this video I had the great pleasure of speaking with Matt Yousefzadeh, a research assistant professor at the University of Minnesota who's made many great contributions to the research field of cellular senescence, DNA damage and aging. I hope you enjoy our conversation. So hi Matt, thank you for joining me today and welcome to the Sheekey Science Show.
Yeah, thanks for having me. I'm really excited about this.
Awesome. So I thought I would start by talking about probably one of your most cited publications, which is about the compound Fisetin or Fisetin or whatever you'd love to pronounce it, where you describe its action as a Senotherapeutics. And so that covers the topic of cellular senescence, which I'm sure my viewers are somewhat familiar with, but maybe a small introduction would be helpful. But I was just wondering if you could provide some background to this work, why were you looking at Fisetin and what exactly were you looking at in the paper?
Mm-hmm. So once again, just the lab that I'm in really focuses on cellular senescence and senolytics, and cellular senescence is cell fate, whereby cellular senescence is induced by stress. Cells basically undergo this cell fate where they're no longer able to proliferate, even if you give them mitogenic stimuli, so you can dump in all the nutrients and growth factors that you want, they're not going to proliferate, grow, and divide anymore. However, they're metabolically active and they can produce some pretty nasty stuff, so inflammatory chemokines and cytokines, and that's known as the senescence-associated secretory phenotype, or SAS, which can then sort of act both proximally and distally to create inflammation, drive secondary senescence, and sort of disrupt tissue homeostasis, and drive aging, where it ultimately shortens health span, disease-free survival, or lifespan. And so right as I joined Laura's lab, the first Senolytics paper came out where it used the senolytic combination of dasatinib and quercetin, and it showed really promising effects, and it's gone on to launch a number of clinical trials. And as we get into making second and third generation senolytics, we want to make more effective drugs, but also safer drugs. So dasatinib is a chemotherapeutic tyrosine kinase inhibitor that has its own sort of known side effects. And so we went and looked at analogs of quercetin, so polyphenol flavonoid, and we screened other flavonoids to see if they were as potent as quercetin or more potent. And what we came up with was we found Fisetin was using an in vitro screening model for eradication of senescent cells. Similarly, we did this both in a human and a mouse cell line. And then from there, moved into some mouse studies where we were able to see using either naturally aged mice or a mouse model of accelerated aging, that's sort of the backbone of our lab. We saw significant reductions in senescent cell burden. And if we did a late life intervention with really old mice, where they're sort of at the human equivalent of about 65 years of age, and you start them on a diet with Fisetin where it's compounded into the chow, it extended lifespan and it reduced pathology as well as senescent cell burden in them. And so that's really kind of where we've started with that. And now we're sort of using Fisetin and a number of other things, where we had another paper that had come out showing that we use Fisetin in a mouse model of normal microbial experience. So most typically, everybody that's doing mouse model work keeps their mice in very pristine conditions, specific pathogen free. And really, we want to better mimic what's going on in the human environment, where we're always exposed to pathogens. And we're very susceptible as we get older to sepsis and other infections. And that's not always seen when you have a really clean environment where you're housing old mice. So we would actually either co-house our mice with pet shop mice, or you would house them in a cage with fomite bedding from those pet shop mice. And we used what we found was the agent that was most lethal to them in the fomite bedding or in the pet shop mouse was mouse hepatitis virus, a beta coronavirus. And typically, we had come up with this hypothesis, this rheostat model, where infection will drive inflammation, but because these older mice had such a higher senescent cell burden, they were already pre-primed for senescence. And so you get this hyperactivation of SASP inflammation, and that can drive mortality and decrease survival. And so what we did was we either tried pre-treatment with Fisetin, or in a therapeutic study, we would induce infection or co-housing or fomite bedding, and then give them Fisetin. And what we saw was a reduction in senescent cell burden, as well as a significant increase in survival.
Wow, no, I didn't know that. That's really interesting results. And so, like, you've mentioned how you give it to the mice, but how exactly are the mice getting the Fisetin? Because obviously, like, in terms of bioavailability, how is that resolved? And like, in terms of like applying it to human clinical trials, like, what do you think would be the most effective like strategy to employ Fisetin?
So in the mouse coronavirus trials, it was given typically one of two ways. They usually get boluses through oral gavage, and then on non-gavaging days, you would give it in the chow. Originally in the paper with the late life intervention, it was always compounded in the chow. It's the easiest way to give it to them. It's the least stressful on an animal. And I think in the human clinical trials with Fisetin, they are given it orally. So.
Okay. And so one of my questions about Fisetin as well was like, obviously as being like the kind of biochemist that I am, I really want to know like the mechanism of action of Fisetin. And so is there any idea as to how it's working yet?
I think Fisetin is one of those compounds that falls into the category of dirty drugs. It's probably doing a ton of different things. There's papers out there that say it stimulates glutathione production. It's involved in inhibiting certain molecular pathway. Some people think it's acting through mTOR. I think we would have to sort of sit down with the individual compounds that can act on those same pathways and see, can we recapitulate the effects of Fisetin and sort of see which one of those molecular targets is actually most important to sort of the Senotherapeutics effects of Fisetin. But it definitely doesn't fit the classical pharma model of one drug, one target.
Yeah, no, for sure. And so, like, obviously, one of the reasons you mentioned why you studied Fisetin was to find like an alternative to the combination of dasatinib and quercetin. But a study that came out at the end of last year identified a new like senolytic agent in grapeseed extracts, procyanidin C1. So, I mean, like, how do you think that compares to Fisetin? I don't know if you've seen the study, but and do you think that Fisetin may be like targeting a specific like subtype of senescent cells, or like could you elaborate on that?
Yeah, so I saw that study. It's really promising. It's cool that we're seeing more Senolytics coming out. Hopefully we're making better ones. I love it if there's something better than Fisetin. It means it has more potency and it's going to help more people out. I think the other thing is, we have to think about that senescent cells, they have these things called SCAPs, or senescent cell anti-apoptotic pathways. And these SCAPs, or these pathways that are upregulated, are probably going to be in some context cell type or tissue specific. And we've seen before that certain senolytics have more of a potent effect in certain cell types. So there is going to be cell type specificity to the effect of some of these Senotherapeutics. And going forward, there may be a senolytic that works better in the kidney than it does in the knee joint for osteoarthritis. So I think that's where the field has to go. We have to learn a lot more about senescent cells. And there is a really interesting grant mechanism that came out that was sponsored by the NIH in the US. And it's having these senescence tissue mapping centers, or what's called SenNet. And so I think there was originally supposed to be 6 of them, but there's now 8 of these tissue mapping centers, University of Minnesota being one of them. And they're mapping senescent cells in particular tissues in healthy aging. And so I believe the U54 tissue mapping center at the University of Minnesota is tasked with liver and adipose tissue. Another institution has brain, somebody has lung, kidney. And similarly, there's going to be the mouse version of this that I think everybody was submitting grants for that were due on Tuesday. And so they'll be matching up to those tissues as well as doing others. And so it's a great way to sort of see what we get in the human side and the mouse side. Obviously, human being the much more relevant, but mice, we have so many tools and so many good models of senescence. And really, you're just trying to build not just a 3D map of senescence. So say in a liver, is it a senescent hepatocyte? Is it a macrophage? Is it an endothelial cell? Are they senescent? What markers are they expressing? And then what about the cells around them? So you're not only creating a 3D map, but a 4D map because you're looking with time.
That'd be really cool to get that data.
Yeah, so these are 5-year initiatives. So I think at the end of the 5 years, we're going to know way more than we know right now.
Yeah, that'd be really cool. Yeah, because as you say, there's senescent cells are very heterogeneous. It depends on their location and also like how they become senescent in the first place. And so just keeping with Senolytics a little bit more, like what are your thoughts on the whole like hit and run strategy approach?
I think the hit and run strategy approach is, that to me it's the smart way to do it. If we basically say we have a drug that eradicates senescent cells, if it's, cleared out the bulk majority of the burden, you shouldn't have to be taking it constantly until you sort of reach another critical mass of senescent cells. Now, they may have beneficial effects, say like a Fisetin, if it is a glutathione stimulator, it could have positive effects for continuous treatment. And, that's some of what we've done with these late life intervention studies is we just kept them on the chow because we don't know the dosing regimen yet. I think everybody would agree in the senolytics field. We don't know, truly the frequency, the dose, everything per person. We're still sort of working that out. But ideally, we would be doing hit and run. And it's probably a way that if you had a senolytic that maybe had some side effects or everything, you're sort of de-risking it and minimizing it by only giving it intermittently.
Yes, okay, that's a really good point. And so also in terms of like senotherapeutic strategies, Senolytics is kind of like just like one branch of the tree of approaches. And like the two other methods are xenomorphics and kind of activation of the immune surveillance. So the immune cells that could maybe clear the senescent cells. Yeah. And so that kind of leads me on to a paper that you published last year about advancing immune system aging and how that seemed to lead to an increase in senescent cells. So could you elaborate on that story?
Yeah, I think the only thing I want to say about that point that you brought up is I think Senotherapeutics is the big umbrella in which everything sort of rests underneath. You have Senolytics, which actively and selectively kill senescent cells. You have xenomorphics, which are drugs that don't kill senescent cells, but maybe they suppress that senescent phenotype. So they're reducing the amount of SAS. And then you're right, you do have drugs that are probably immune stimulatory that basically bring in immunosurveillance and the eradication of senescent cells that would typically occur like in a young and healthy individual. And that's really important that basically there's this concept called immunosenescence, which is different than, say, senescence in immune cells. Immunosenescence is the progressive and age-related decline in immune function. It's marked by a number of things: thymic atrophy, reduced T cell response, stem cell exhaustion, increased markers of immune exhaustion. And so what we wanted to do is investigate the importance of the immune system and senescence in the immune system on the rest of the organism. And really, by and large, before this, what we've done in Laura's lab was we've worked with systemic models of senescence where we knocked out a DNA repair gene, ERCC1, throughout the mouse, and we saw a significant increase in senescence throughout the entire mouse. That's great, but if you want to study segmental aging, or I want to see what happens in the spleen and then what the spleen does to the rest of the body, the cell non-autonomous effects, that model's not so great. So we made tissue-specific knockouts of ERCC1 using the LOXP-CRE system, which is a really nice system that everybody uses to delete genes in a sort of temporal spatial manner. And so we deleted ERCC1 in hematopoietic stem cells using the CRE recombinase driven by the VAV promoter. And so we, in effect, knocked out ERCC1 from hematopoietic cells. And what we saw was that when the mice are young, there's no increase in senescence in the lymphoid organs. But as they get older, as they get to about that 5 or 6 month stage, we start to see senescence and dysfunction there. And it really hyper elevates as the mice get older, so that the level of senescence in a spleen by about a little less than a year looks like that in a 2.5 year old mouse. Similarly, if you can measure immune function in those mice, like you can do foot pad swelling, where you sensitize them to an antigen and then look for foot pad swelling when you sort of give that antigen back, similar to how you would do a TB test, where you're looking for the mark on your arm. We saw significant reductions in that mouse model, and that reduction was comparable to, but didn't exceed the level that was observed in an old mouse. And going back to your point of immune surveillance, we also looked at NK cell cytotoxicity. So NK cells, T cells, macrophages are all thought to aid in senescent cell removal. And some of the work that's been done by Valerie Krzyzewski's lab has really shown that NK cells can play a prominent role in this. And so following upon that line and sort of using his work as inspiration, I measured NK cell cytotoxicity from these immunosenescent mice and saw a significant reduction in their cytotoxicity, showing that, there could be impairments in the clearance of senescent cells that occur through the increased endogenous DNA damage, which drives senescence in the immune compartment.
Yeah, that study, when I read it last year, was so interesting, and it kind of raises the idea of like maybe a vicious cycle, that obviously you age the immune system, and then that can also mean that you reduce the clearance of senescent cells, which then could cause more aging. And I think it's really fascinating.
Yeah, I sort of use the analogy that the immune system is making a bunch of trash through increased SAS, but then the immune system itself is tasked with clearing the trash out, and it's not even doing a good job at that. So the whole house is just filling up with rubbish.
I think you've just reminded me, this is a very neat reference to Monsters, Inc., where there's like one of the monsters is cleaning up the floor, but it's a snail, so it's also like creating more like slime behind it. But that's besides the point. One thing else that I found very interesting with your paper is that as you, I think you've already mentioned that as you like reduce the function of the immune cells, you also saw senescent cells increase in different organs that weren't immune system organs. And I believe you saw like in particular increase in senescent cells in like the liver and the lung, but maybe not so much in the skin. And so I was just wondering if maybe this data could infer where immune senescent surveillance is more important.
So I think you could infer some of that. I think that it's the caveat to it. I should sort of work backwards and say that we were looking for cell non-autonomous effects, and what we saw was senescence occurring in the non-lymphoid compartment, so solid organs. And the first question there is, well, is it just resident immune cells in those tissues that are senescent? And it turned out not to be. It turned out to be bona fide parenchymal senescence, because we did co-staining and in situ hybridization with one of our collaborators at the Mayo Clinic. And so it really is senescence occurring in those non-lymphoid organs. We're seeing the resulting tissue dysfunction, so increased markers of liver dysfunction that are observed in the blood. We saw increased proteinuria in the kidney, indicating kidney dysfunction. Now, as far as going back where the tissues had the highest level of senescence versus lower ones, I think some of that is also seen naturally when we looked at naturally aged mice and systemically aged accelerated aging mice, where like the aorta, for instance, when we looked at naturally aging and the accelerated aging models, had the highest sort of ratio of senescence in the old mice compared to the young mice. And that just happened to sort of bear play. Now, I don't know if that's just those tissues have more of a propensity for senescence. They're more sensitive to it, and they have much more of a dynamic range that's there. Or does immunosurveillance also come into play? And that definitely could be the case. I think that's going to take a lot more study.
Yeah, for sure. Because like, I guess, do you think that certain organs are more accessible to the immune system, and so immune surveillance is more likely in those organs?
I definitely think so. I think, where the immune system can go, say, would you take something like the liver, which is rich in immune cells, versus, say, an immune privileged organ like the eye? I think that would be a cool sort of study to look at there, and seeing, the effects of defective or immunosenescence mouse model, and what are the effects on the eye. Unfortunately, the VAV model doesn't work so great because there is leakiness in the eye of the mice. So we would also be deleting the target gene there too. So it really kind of muddies the water.
Yeah, no, I think these questions are really interesting, and hopefully, we can develop like experimental methods to try and address them. But like just one, the other thing that you did in that paper was you then treated the mice with rapamycin, I believe.
Yeah.
And could you explain that part?
Yeah. So rapamycin, basically compound found produced by the bacteria on the island of Rapa Nui or Easter Island. It's been shown, I think for about 13 years ago, was shown to increase lifespan in mice as part of the NIA's intervention testing program. Some other people had shown it was immune stimulatory in mice, and Joan Manick, when she originally starting at Novartis and at Restore Bio, had papers out showing that it was immune stimulatory in elderly individuals, where they tested them with a short course of rapamycin treatment and then looked at influenza vaccination titer levels and saw a 20% increase, which is massive considering how poor vaccination efficacy is in the elderly. I think they also saw reduced PD-1, a marker of immune dysfunction and exhaustion, on T cells as well. So sort of drawing upon inspiration from that, I wanted to see, could we use the mouse model possibly as a vaccination model? Or would rapamycin just be immune stimulatory? Could it correct some of the deficits? And, giving the mice 6 weeks of rapamycin before letting them sort of wash out, and then performing some of the analysis of immune function, we did see a partial correction of deficits. It wasn't a full restoration, but it definitely made things better. It reduced systemic senescence, it reduced the circulating SAS factors, and helped to improve some of the lymphopenia that we observed in the mutant model.
Okay, that's really interesting. And so, like rapamycin, I believe, because of the way it inhibits mTOR, and mTOR being involved in the SASP also seems to maybe be acting as a xenomorphic. Like, would you agree with that?
Mm-hmm. I would definitely call rapamycin a xenomorphic at least. So yeah.
And so I think, the other thing, so we've spoken a lot about, I guess, like immune surveillance and senolytics, but in terms of the xenomorphics, like sometimes I hear it described as like complete blunting of the SASP, but I guess I think the better way to kind of describe it is there's like good and bad secretory components of the SASP. And so I guess the xenomorphics in theory would be a way of trying more to suppress like the bad aspects as opposed to the good aspects.
I think that's right, where there are positive aspects of SAS, and there's positive aspects of senescence. Senescence is incredibly important for developmental biology, where you needed a cell population for development, then you need to shut it off and get rid of it. And the way it gets rid of it is by producing the SAS to come in and eradicate those cells. Similarly, it's important for wound healing too, to ultimately prevent excessive fibrosis in a young, healthy system. Now, as we get older, we have that sterile increase in inflammation and what's known as inflammaging. So I think that, the xenomorphics, it depends. I think Thomas von Zienicki's lab has shown that maybe aspirin and other NSAIDs could even be xenomorphic. So I guess it depends upon the degree of activity, and like everything else, there's always a therapeutic window where you have, a low dose, it's not really that effective. You have that nice sort of sweet spot, and above it, you have, toxicity or unintended off-target effects.
Yeah. And so kind of segueing a little bit, one topic that I also think is kind of important in the field is like, what is the main inducer of senescence in vivo? And I know that you've done a lot of work with looking at DNA damage. And so I was interested to hear your thoughts on, what you think the answer might be.
So my background's on DNA repair, so I'm partially biased. I think I would take a beating from everybody if I said DNA damage is the driver of aging. So I will be cautious and say it is a driver of aging. It's, genomic instability is one of those hallmarks of aging where you have all of them. And, there's so much interplay between them. DNA damage can drive senescence. Senescence can bring in inflammation, which can in turn drive DNA damage. So everything's interconnected. There's, forward feedback cycles. everything. But what we do know is that we've looked. Laura, before I got to the lab, had profiled tissues for certain DNA damage types and certain lesions. One of them being oxidative lesions, these cyclopurine adducts. And what she had saw in certain tissues, they increased with age, but certain ones they didn't. Like they increased in the liver and the kidney, but not say in the brain or the heart. And it could be that certain tissues have this threshold for DNA damage where if it reaches a threshold, now there's stress, and then they can invoke cellular senescence. Whereas that second threshold in certain tissues, like the brain, may be very low, where you have stress, and then you just have apoptosis. And there's very little room for senescence there in certain cell types. I'm sure, it's organ dependent, but it's also cell type dependent. And that may be why we didn't see those increased lesions in certain organs, is because those cells were just driven to apoptosis.
Yeah.
Or other cell death mechanisms, I should say. So kind of moving on beyond what I said before about DNA damage in tissues, we've also learned a lot from aging from individuals that were treated with cancer, where chemotherapeutics, we know in some cases, can prematurely drive frailty. So that a pediatric cancer survivor, by the time they hit their early 30s, if they've experienced multiple rounds of chemotherapeutics, they could end up looking like a 60-year-old, based upon frailty scores. We also have these DNA repair disorders where they have features of aging, whether it be systemic or tissue related. And some of them, it gets further complicated by them displaying mosaicism as well. But the mouse model that we work on, the ERCC1 model, is based off of XFE progeria, which was the sentinel patient had a mutation in XPF, the binding partner of ERCC1. And it's basically a molecular pair of scissors that's used to clip out damaged DNA or just overhangs that need to be trimmed away. And if you think about it, like XPF is the blade of the scissor and ERCC1 is the handle, which binds onto the DNA. So there's a lot that we've learned from the rare disease patient space, as well as cancer survivors, and the effects of DNA damage on aging as well.
That's really interesting. And so like one thing I think is worth mentioning is like, when I think about DNA damage, there's kind of like two main categories. There's like exogenous damage mechanisms and endogenous damage. And like, so could you elaborate on like what are the major differences between them, and which one do you think is more likely to be causing aging in the body?
Yeah, so exogenous sort of occurring outside the body, endogenous occurring inside of it. Exogenous would be you go out in the sun for the entire day, and the next day you're as red as a lobster because you've incurred so much UV photo damage from being outside. And there's a rare disease group that we work with called xeroderma pigmentosum, or XP. And so these patients have mutations in the nucleotide excision repair, of which ERCC1 XPF plays a role in, that are used to remove photo lesions as well as bulky DNA adducts. So things that occur like BPD adducts that occur from cigarette smoking. And endogenous could occur from things such as adoxoguanine that occur from oxidative damage that arises from normal metabolic processes. Obviously, the more dysfunction you have going on, say as you get older, the more mitochondrial dysfunction, the more reactive oxygen species leak you're going to have, which can in turn increase more of these oxidative DNA lesions. But just typically, I think the estimates are per cell per day, you have 50,000 DNA damage events. Wow. So there's already this baseline that's going on in every single cell per day. And obviously there's variability between the different cells in your body where, oxidative DNA damage may be more common in the liver, say bone, where normoxy in a bone is 0%.
I see. And so like, in terms of the fact that obviously there's like endogenous repair mechanisms within our cells, do you think it's possible that we could like try and activate those to try and alleviate some of the like increased damage that we seem to see with aging?
Yeah, and for the most part, obviously loss of these repair mechanisms is pretty deleterious. Not really, most of these repair mechanisms, you can't just add back one factor or an excess of one factor and it fixes everything. I know people are working with CERT6, and CERT6 is involved in a number of different repair pathways, as well as other processes. It's got a couple of different enzymatic functions. So I think it's not going to be that you could just throw in this one enzyme and it's going to fix everything. However, there are compounds that are stimulatory for repair processes, and maybe these might actually work. So that may be a possible therapeutic.
I see. And so besides obviously DNA damage, what other like major factors do you think are causing aging?
So I've always gotten interested in oxidative stress. That was kind of one of the first projects I worked on when I joined Laura's lab. And what we saw is that as you get older, your body is producing more reactive oxygen species. You're also simultaneously, you're losing your antioxidant buffering capacity. So enzymatically, catalase activity is down, as well as the expression of some of these antioxidants. So it really creates the vicious cycle. So that's kind of one of the other things I've been interested in. And now more and more looking at the role of the immune system in senescence, both driving it and from a surveillance standpoint.
So are you able to like elaborate a bit more on like your current research projects?
Yeah. So moving beyond that immunosenescence mouse model, we kind of wanted to look at, well, the different immune cell populations. Does it take all of them in concert having dysfunction driven by deletion of ERCC1 and increased senescent cell burden to really drive organismal aging? Or is one cell type more prominent than the other? So we're trying to make different immune cell type knockout. So a B cell knockout, a macrophage knockout, and going from there. There was a preprint out by George Garinas' lab that showed CX3CR1 Cree mice where they deleted ERCC1. So it can delete in tissue resident macrophages as well as microglia. The mice had cerebral atrophy. I don't know that they've done any lifespan analysis, but does seem to be some systemic effects going on too. So I think this is going to be a pretty promising thing to look at. And say if you find something in a T cell, then you've really got to stratify down even further. Well, what about a CD4 T cell, a CD8 and effector T cell, regulatory T cells? Somebody can make an entire career off this. So.
No, I agree. The immunology is was complicated, and I guess it's hard to to understand the different like categories of different immune cells and understand like the receptors and how they interact. So I guess another interest of mine is like, how are immune cells recognizing senescent cells? Is it purely based on something that they're secreting, or like, what are your thoughts on that?
Sure. Well, I think there was the paper that came out from Judy Campisi's lab with NKD2G, which is a signal for NK cells and to sort of eat things or engulf things. I believe I'm wrong. Somebody correct me, but I'm only on here with you right now, so we'll go with it. And what the senescent cells can do is part of the SAS, they can produce those or express those matrix metalloprotease enzymes, which could then cleave that marker off the outside of the cell. And I think it was done through MMP3, 6, or 9. And so you're sort of losing that signal that allows them to sort of be eaten and degraded. So that's part of it is, the cell surface that things are decorating themselves with to track the immune cells. They're sort of trying to clip them off, don't find me. But then also as you get older, those immune cells just aren't doing their job as well either.
Yeah, and it also just reminds me of another interesting avenue of like trying to clear senescent cells, which is like the CAR T cells that are like designed to clear senescent cells by targeting like proteins that are only expressed on the surface of senescent cells and also not the healthy cells, as a way of being more specific than using like different senolytic drugs.
And so I think they're talking about specifically the work that was done by Scott Lowe's lab. And I think right now we're hampered by that there's not really a great specific senescent cell surface marker. But you could get around that in some cases by having CAR T cells that have low affinity, right? So if you have this marker that's really high on senescent cells, but it's also found on other cells, you would use a CAR T that has a low affinity antibody. So it's really only latching on to senescent cells and eradicating them. And so that could be a technique for that. So it all depends upon the target discrimination. And then with CAR T, you can play with antibody affinity as well.
That's a good point. I didn't think of that. So I think the senescence field is really exciting. Obviously, I think we're both biased because we're both in the fields, but I think there's lots of different approaches, and there's so many questions like still left to address. And so I guess you go.
I agree with you that we're biased. I think there's so much cool stuff that's going on in the aging space, and that you're really cool if you work in it, but you're cool if you work on senescence.
Even cooler, yeah. And so you've spoken a lot about senescence now, but what other areas of the aging fields are like kind of are of interest to you?
So really, I'm heavily into senescence and DNA damage. The other thing that I've sort of gotten into over the last 5 or 6 years working in Laura's lab is actually outreach. Okay. So we have been doing these workshops for underrepresented groups to bring them into the aging field, to bring the aging field to them, not only giving them didactic lectures from experts in the field, also giving them some laboratory experience, really an exposure, and then going from there to help sort of get them and keep them in the pipeline. So I've been working on that and more helping to do some mentorship in the lab. And, because I can only do so much at the bench, but if you can take and train and, inspire people and hopefully, you've enabled and empowered them, they can drive the community so much further than I can ever do on my own. And that's kind of what I've been trying to work on. And recently I've been, I've sort of taken on the role as the geroscience junior trainee reviewer, sort of the chief reviewer, to get trainees for the American Aging Association, sort of at that senior graduate student and postdoc level, get them into the pipeline and get them starting to review papers as they're really starting to write their own, or they have been writing their own, to, maybe that helps them increase the quality of their paper, or maybe it just helps them to become a better reviewer. I'm satisfied with that.
That sounds that's really important work. And so, I mean, what kind of advice would you give to people who are like either interested in the field or like, I guess, researchers like myself?
Just start building a network and emailing people. Even if you think you have no business talking to them, just send them an email. Hey, I saw your paper. I really liked it. Like, I really liked your review article. I thought it was great. I think everybody emphasizes P16 and P21. I thought your review article did a great job of reminding everybody about P53 and, the non-stress state, the acute state, and really the chronic state, and, things that we need to think about. I think it's sort of underthought of.
Thank you.
Yeah. So I think it's just things like that, just sort of meeting. If you can get to a meeting, meet as many people as you can. Even now we've gone so virtual, just start emailing people. Can I have an informational interview? What do you do? What's your day like? What projects do you have going on? What advice do you have? And most people, they can carve out 20 minutes to chat with you.
Yeah, no, I completely agree. I think, as you say, yeah, I know as well you're very prevalent on Twitter. And I think, again, that's something I try and like advise people interested in science to do, because it's a great way to just see like the current research papers.
Yeah, I would think that you and I probably, just by being on Twitter, probably have immediate access to those papers before we ever would have seen them on PubMed or through other searches. So I feel like compared to other people in the lab, I'm a couple of days to a week ahead of them on papers.
Yeah, exactly. I mean, that's one of the main reasons why I use it. And given speaking of like recent announcements, there was the announcements of Altos Labs today. And so I guess on from that, like, what are your just general thoughts about the different companies that are now rising in the field? And I guess your thoughts on cellular reprogramming?
I think cellular reprogramming is really interesting. I think it's, once again, it's gene therapy. So there's an uphill sort of struggle with it. I mean, can we find small molecules or biologicals that mimic the biological benefit of epigenetic reprogram? I think that's really how you democratize the science and the therapeutics. Can we basically do that to give cellular reprogramming to the developing world so that it's not just a first world treatment? That it could be used in a rural clinic somewhere. I think now is probably the best time to ever get into the longevity space, as far as from an academic standpoint, as well as even from a commercial standpoint, I'm also sort of looking to get into that. Is there anything that I can do to help to commercialize some of the discoveries that we've made in the lab? And so I'm currently doing the On Deck Longevity Biotech Fellowship Program, and I've enjoyed it. I've made a lot of great connections, a lot of people, both from the investment standpoint, operations, as well as, academics that are looking to sort of get into that or just go work for a startup. It's been great. I think they're recruiting for cohort 2 right now, so definitely recommend everybody to do that. But yeah, there's no better time to get into this. And you saw Altos, I guess it's no longer, can you call it a startup when it's a $3 billion, backing to the company, but they've assembled a dream team. They're flush with the war chest. It's going to be a struggle, but I don't think anyone else is capable of doing it as well as they can. So
yeah, I think it's going to be really exciting to see where they go with it. And so I guess a final sort of question is, what do you think are the biggest unanswered questions in the aging field at the moment?
I'm going from a very senescence-centric worldview. It's just my worldview. It's not the right worldview, but cool if you share that worldview. So I think, and that's what, getting back to those tissue mapping centers is, where are the senescence? What are they? What's around them? We're still building out good tools to see and visualize senescent cells, and what are they doing to the cells around them? And so that's things where we're utilizing emerging technologies like mass imaging, spatial genomics. I think, that we're using such primitive techniques to identify senescent cells. And I really think we're building the next generation tools right now, whether it be spatial genomics or people like Nate Basisti, who used to be at the Buck Institute, that's now at NIA, looking at the secretome and SASP senescent cells. That's their, building better Senotherapeutics, whether it's Senolytics, Xenomorphics, Senostatics, whatever we want to call the new drug class. How do we do precision Senotherapeutics, whether they're more effective, less toxicity, more cell type specificity, and then you can start putting them together in combinations. I think that's kind of the future of the field that really excites me.
Yeah, no, me too. I have some personal interest in the SASP composition for my own research project. So yeah, I'm totally excited for the field. Thank you so much for giving your insight to the field today. Where can my viewers go to hear more about your work or to kind of follow more about you?
I'm available by email. I think you can find me online. My Twitter profile is Matt Yousefzadeh. I love to chat about science. I love bad puns. You got memes, that's great. So, but yeah, just find me and engage with me, and send me a link that you think is cool. I'll definitely read it.
Awesome. Yeah, so thank you again for coming on today. Best of luck for your research avenues. Yeah. I'm sure we'll hopefully meet at some point in person, given our overlapping interests. So yeah, thank you again.
Thanks for having me.
So I hope you've learned something in this video. Thank you to my Patreon supporters, and thank you for listening.